Right now, there's a biological barrier protecting you from flesh-eating flies, and it's made entirely of sterile insects. This isn't natural. It's one of the most ambitious scientific interventions in history, requiring the weekly release of millions of radiation-sterilized flies across multiple countries. Imagine entire herds in Texas spared the gruesome fate that once killed 500,000 cattle in just one outbreak. This scientific intervention has never stopped since the 1950s. Over 100 million sterile flies still stream from the sky each week to hold back a lethal parasite. But here's what makes this even more incredible. It's the only thing preventing a catastrophic parasite invasion that would devastate livestock across entire continents. The invisible army above your head. Aircraft drop these sterile males across Panama, Costa Rica, Nicaragua, Honduras, Guatemala, and southern Mexico. Small boxes of sterile male flies pop open in flight, scattering thousands across a grid of fields the size of small countries. The coordination required is staggering. Multiple countries synchronizing their releases to create overlapping zones of sterile fly coverage. Weather conditions, wind patterns, and seasonal breeding cycles all factor into deployment schedules that must be maintained week after week, year after year. This invisible shield works because sterile males compete with wild males for mates, but when females choose the sterile males, because a female screwworm mates just once before dying, a single sterile mating spells doom for that entire genetic line. The sheer volume of sterile flies creates overwhelming odds against successful reproduction by wild populations. In areas where the program operates effectively, sterile males can outnumber wild males by ratios of 100 to 1 or higher. The female flies have no way to distinguish between fertile and sterile males, so they mate with whatever males they encounter first. Most people walking through these protected territories have no idea they're surrounded by millions of deliberately sterilized insects. The flies look identical to their wild counterparts. Same flight patterns, same mating behaviors. The only difference is their inability to produce viable offspring. This biological masquerade is essential to the program's success because the sterile flies must remain sexually competitive to effectively intercept wild females. If this protection disappeared overnight, the consequences would unfold rapidly. Wild screwworm populations would explode within months, with females laying hundreds of eggs in livestock wounds. The parasites would spread northward through Mexico into Texas, Arizona, New Mexico, and California. Ranchers would face the same devastating losses their predecessors experienced before the program began. Hospital emergency rooms would start seeing cases of human myiasis, screwworm larvae eating flesh in wounds that seem minor at first. The program essentially wages biological warfare using the enemy's own species against itself. Instead of trying to kill screwworms with pesticides or barriers, scientists learned to create an army of reproductive saboteurs. These sterile flies carry the same instincts and behaviors as their wild relatives, but their biological weapons programmed to end genetic lines rather than continue them. The strategy exploits the screwworm's mating behavior as a vulnerability rather than fighting their physical presence directly. This weaponization of sterility required decades of research to perfect. Scientists had to determine optimal radiation doses that would sterilize males without making them too weak or behaviorally abnormal to compete for mates. Too much radiation would kill the flies or make them unfit for reproduction. Too little would leave them partially fertile, potentially creating resistant populations. The precise calibration took years of experimentation and field testing to achieve. Modern mass rearing facilities now produce 100 million flies weekly using precisely calibrated diets and radiation. Advanced genetic screening ensures only the healthiest flies enter the sterilization process. Quality control systems test samples from each batch to verify sterility rates and meet operational standards. Current facilities can process millions of flies simultaneously through industrial-scale radiation chambers. The logistics of moving this many insects from breeding rooms to sterilization chambers to packaging systems requires assembly line efficiency. Specialized containers maintain optimal temperature and humidity during transport to aircraft. Loading systems transfer millions of flies into aircraft dispensers within tight time frames to maintain fly viability. 7 decades of continuous operation means this program has never experienced a complete shutdown. Even during political upheavals, natural disasters, and global pandemics, the sterile fly factories kept running. Aircraft kept flying their missions. The program survived changes in government, shifts in scientific leadership, and multiple reorganizations of participating agencies. This consistency is crucial because any significant gap in deployment would allow wild populations to reestablish themselves. The dedication required to maintain this schedule is extraordinary. Breeding facilities operate around the clock, with staff working shifts to monitor insect health, adjust feeding schedules, and maintain optimal environmental conditions. Pilots fly predetermined routes regardless of weather conditions that would ground normal aircraft. Ground crews load aircraft in pre-dawn darkness to ensure timely deployment. Laboratory technicians process quality control samples within hours of collection. Climate change is now forcing program expansion beyond its traditional boundaries. Rising temperatures are creating suitable screwworm habitat in regions previously protected by cold winters. Northern Mexico and southern Texas are seeing extended breeding seasons that require year-round rather than seasonal deployment. New territories in South America need protection as warming temperatures make previously marginal habitats suitable for screwworm establishment. The program's response to climate change requires additional aircraft, expanded breeding facilities, and larger staff. Scientists are developing predictive models to identify areas likely to need protection before screwworm populations become established. Early intervention requires deploying sterile flies into areas where wild populations don't yet exist, essentially creating preemptive biological barriers. Recent expansion into Libya and other parts of North Africa demonstrates how quickly the program can mobilize when screwworm populations appear in new territories. These emergency deployments require massive resource allocation and international coordination. Aircraft must be positioned, facilities must be established, and local staff must be trained within months rather than years. The program's success depends on maintaining sterile fly production at levels that consistently overwhelm wild populations. Any reduction in deployment numbers creates opportunities for wild flies to find mates and reproduce successfully. The mathematics are unforgiving. If sterile males don't maintain overwhelming numerical superiority, the biological shield begins to fail. Current deployment schedules account for seasonal variations in wild fly populations, adjusting release numbers based on breeding cycle predictions. Summer months typically require higher deployment rates as wild populations peak. Winter deployments maintain baseline protection during periods of reduced wild fly activity, where delays can create gaps in coverage that must be compensated by increased releases once conditions improve. The program represents one of the largest coordinated biological interventions in history. Multiple countries synchronize their activities to create continental-scale protection. Research facilities share data on fly genetics, breeding techniques, and deployment strategies. International agreements ensure continued cooperation regardless of political changes within participating nations. This protection requires constant vigilance because screwworm populations can reestablish quickly if sterile fly deployment falters. The program's success creates a dependency that can never be abandoned without risking catastrophic consequences. Every week's deployment is essential to maintaining the biological barrier that protects millions of animals and humans from flesh-eating parasites. The massive resources invested in this program reflect the enormous economic and humanitarian stakes involved. The alternative, allowing screwworm populations to reestablish, would cause suffering and economic losses that dwarf the program's operational costs. This invisible army of sterile flies represents humanity's commitment to protecting itself and its animals from one of nature's most destructive parasites. But what exactly makes this enemy so devastating that it requires such an extraordinary response? The answer lies in understanding the biological machine that nearly brought livestock farming to its knees. When paradise became hell. Cochleomia hominivrax operates as a biological machine designed for one purpose, converting living flesh into more screwworm flies. These metallic blue-green insects with bright orange eyes possess sensory capabilities that make them among nature's most efficient parasites. Their antennae can detect the chemical signature of an open wound from distances exceeding 2 miles. Blood proteins, tissue damage markers, and bacterial compounds from infected wounds create a scent trail that draws female screwworms like a beacon. Once they lock onto a scent, they can track it across vast territories, navigating through dense forests and open grasslands with remarkable precision. The female screwworm approaches a wound with surgical precision, evaluating the site for optimal egg placement. She prefers fresh wounds with exposed tissue and adequate moisture to support larval development. Using her specialized ovipositor, she deposits clusters of cream-colored eggs directly into the womb cavity. Each female can lay up to 400 eggs in a single session, and she will repeat this process multiple times throughout her 3-week lifespan. The eggs attach firmly to wound tissue, making removal nearly impossible without causing additional damage to the host animal. Within 24 hours, the eggs hatch into tiny larvae equipped with powerful mandibles designed for cutting through living tissue. These larvae immediately begin feeding on healthy flesh surrounding the wound, secreting enzymes that break down proteins and prevent blood clotting. The feeding process keeps wounds open and expanding, creating perfect conditions for additional egg laying by other females. Each larva grows rapidly, molding through 3 distinct stages before reaching maturity. The feeding larvae emit chemical signals that attract more female flies, turning a single small wound into a massive infestation site. The expanding wound becomes a festering crater as hundreds of larvae consume healthy tissue. The host animal experiences intense pain and becomes increasingly weak as the wound grows larger and more infected. Secondary bacterial infections often develop, adding another layer of suffering to the already severe tissue damage. In severe cases, the larvae consume so much tissue that vital organs become exposed. Animals can die from blood loss, infection, or organ failure within days of initial infestation. Even animals that survive often suffer permanent disfigurement and reduced mobility from the extensive tissue damage. When livestock farming expanded across the American Southwest in the late 1800s, ranchers unknowingly created ideal conditions for screwworm population explosions. Millions of cattle, sheep, and goats concentrated in relatively small areas provided an abundant supply of potential hosts. Barbed wire fencing caused frequent cuts and scrapes as animals moved through pastures. Branding, castration, and dehorning procedures created fresh wounds that attracted screwworms from miles away. Newborn animals were especially vulnerable, with their umbilical cords providing perfect entry points for egg-laying females. The dense livestock populations allowed screwworm numbers to multiply exponentially. A single infected animal could produce thousands of adult flies within weeks. These new flies would then spread throughout the area, seeking fresh wounds on other animals. The cycle repeated continuously, with screwworm populations doubling and redoubling until entire regions became infested. Ranchers reported losing 30 to 50% of their newborn calves to screwworm attacks. Adult animals suffered constant reinfection, making normal ranching operations nearly impossible. Economic losses from screwworm infestations reached catastrophic levels by the 1930s. The United States livestock industry was losing an estimated $20 million annually to screwworm damage, equivalent to hundreds of millions in today's currency. Texas alone reported losses exceeding $5 million per year. Ranchers faced impossible choices: abandon livestock operations entirely or watch their animals suffer horrible deaths. Some operations went bankrupt after losing entire herds to screwworm attacks. The parasite was essentially destroying the economic foundation of rural communities across the Southwest. Traditional pest control methods proved completely ineffective against screwworms. Pesticides designed for external parasites couldn't reach larvae buried deep inside wound tissue. Spraying animals with insecticides had no impact on eggs already deposited in wounds. Even if pesticides could kill adult flies, new ones would migrate in from surrounding areas within days. The larva's protected position inside living tissue made them virtually immune to chemical treatment. Topical wound treatments might kill surface larvae, but deeper infestations continued developing regardless. Ranchers attempted various desperate measures to protect their animals. They built elaborate screening systems around birthing areas, but flies could still penetrate most barriers. Some tried keeping animals in enclosed barns, but this was impractical for large herds and did prevent wounds from other causes. Others employed full-time workers to inspect animals daily and treat wounds immediately, but this approach was both expensive and ineffective. Even minor delays in wound treatment could allow screwworms to establish infestations that became impossible to control. The screwworm's life cycle made conventional control strategies worthless. Unlike other agricultural pests that could be controlled through habitat modification or breeding resistant crops, screwworms required only wounded animals to complete their development. Every animal injury, no matter how minor, became a potential breeding site. Preventing all animal injuries was impossible in normal ranching operations. The parasite's mobility allowed it to spread rapidly between farms and across vast distances, making localized control efforts futile. Veterinary treatments for screwworm infestations were primitive and often ineffective. Manually removing larvae from wounds was time-consuming and caused additional trauma to already suffering animals. Chemical treatments that killed larvae often damaged surrounding healthy tissue. Many infested animals died from the treatment itself rather than the original infestation. Even successful treatments didn't prevent reinfection, as new flies would target the healing wounds within days. The escalating crisis demanded solutions that went beyond traditional pest management approaches. Screwworm populations were expanding their range, moving into previously unaffected areas as livestock operations spread. The parasite's efficient reproduction and dispersal capabilities made it nearly impossible to contain using conventional methods. Each breeding cycle produced more flies capable of traveling greater distances to find new hosts. The problem was becoming continental in scope, threatening livestock operations throughout North America. Government agencies and agricultural organizations recognized that incremental improvements to existing control methods would never solve the screwworm problem. The parasite's biological advantages were too significant to overcome through minor adjustments to current practices. The situation required a fundamentally different approach that attacked the screwworm's reproductive cycle, rather than trying to kill individual flies or larvae. Scientists needed to find a way to turn the screwworm's own biology against itself. The realization that conventional weapons were powerless against screwworms forced researchers to consider radical alternatives. Chemical pesticides, physical barriers, biological predators, and habitat modification had all failed to provide meaningful control. The screwworm's protected larval stage and mobile adult stage made it essentially invulnerable to traditional attack methods. Only by disrupting the reproductive process itself could scientists hope to gain an advantage. But the solution they would propose sounded more like science fiction than serious research. The nuclear solution nobody believed. Edward Knipling and Raymond Bushland proposed bombarding millions of screwworm flies with gamma radiation to make them sterile, then releasing them into the wild to mate with wild females and produce no offspring. The entomology establishment dismissed this idea as fantasy. Leading scientists argued that radiation would either kill the flies outright or damage them so severely they couldn't compete with healthy wild males for mates. The concept violated everything researchers understood about pest control, which focused on killing insects rather than manipulating their reproduction. The scientific community's resistance was understandable given the era's limited knowledge of radiation biology. Most radiation research in the 1950s dealt with high doses that caused immediate death or severe mutations. The idea that precise radiation exposure could sterilize without killing seemed impossible to most researchers. Critics pointed out that insects exposed to enough radiation to sterilize them would lose their vigor, making them worthless as competitors for wild females. Others argued that even if sterilization worked, producing enough sterile flies to overwhelm wild populations would be logistically impossible. Knipling and Bushland discovered this approach might work because screwworm reproduction depends on a single mating event. Female screwworms mate only once during their 3-week lifespan, storing sperm to fertilize multiple egg batches. If a female mated with a sterile male, she would waste her entire reproductive potential. This biological constraint created a vulnerability that radiation could exploit. Male screwworms produce sperm continuously, making their reproductive cells more susceptible to radiation damage than other body systems. The key insight was that radiation affects rapidly dividing cells first, particularly sperm-producing cells in insect testes. Lower radiation doses could destroy reproductive capacity while leaving other cellular functions intact. The scientists theorized that sterile males would retain normal behavior, flight capability, and sexual competitiveness while becoming reproductively useless. This selective sterilization would turn the males into biological weapons that looked and acted like normal flies but carried reproductive death. Initial laboratory experiments focused on determining radiation doses that achieved complete sterilization without compromising male fitness. Researchers exposed male screwworms to various gamma radiation levels, then tested their mating behavior and sperm viability. Too little radiation left males partially fertile, which could lead to resistant populations. Too much radiation killed flies or made them too weak to mate successfully. The optimal dose required precise calibration measured in thousands of rads. Early tests revealed that males exposed to 7,500 rads of gamma radiation became completely sterile while maintaining normal lifespan and mating behavior. These sterile males competed successfully with normal males for female attention. Laboratory studies confirmed that females mated with sterile males produced no viable offspring. More importantly, the sterile males showed no behavioral changes that would make them less attractive to females. Their flight patterns, pheromone production, and courtship behaviors remained identical to fertile males. The first field test occurred on Sanibel Island, Florida, in 1951. Researchers released sterile male screwworms into a small, isolated population to test whether the technique would work in natural conditions. The island's limited size made it perfect for monitoring results without risking contamination of mainland populations. Within months, the wild screwworm population on Sanibel Island collapsed as females mated with sterile males and produced no offspring. The test proved that laboratory results translated to field conditions. Success on Sanibel Island led to larger trials on Curaçao, a Caribbean island with established screwworm populations. The Curaçao test required producing thousands of sterile flies weekly, forcing researchers to develop industrial-scale breeding methods. They built facilities that could rear screwworm larvae on artificial diets, manage adult fly colonies, and process males through radiation chambers. The logistics were staggering compared to laboratory-scale operations, requiring coordination between breeding specialists, radiation technicians, and field deployment teams. The Curaçao experiment succeeded beyond expectations. Researchers released approximately 400 sterile males per square mile weekly for several months. Wild screwworm populations disappeared entirely within 6 months, with no evidence of surviving breeding pairs. The success demonstrated that sterile insect technique could eliminate established populations from large areas. However, the Curaçao operation also revealed the massive infrastructure needed for continental-scale deployment. Scaling up to protect entire countries would require breeding facilities capable of producing millions of flies weekly. Each facility needed climate-controlled rearing rooms, artificial diet production systems, adult fly management areas, and industrial radiation chambers. The flies had to be reared on standardized diets that produce healthy, competitive males. Adult flies required specific temperature, humidity, and lighting conditions to maintain reproductive behavior. The radiation sterilization process needed precise timing to treat flies at optimal ages for maximum effectiveness. The breeding process began with maintaining colonies of adult flies in large cages supplied with protein-rich food sources. Females laid eggs on artificial substrates that simulated womb conditions. Workers collected eggs daily and transferred them to rearing containers filled with nutrient-rich media. Larvae developed through 3 stages before pupating in sterile sand. Adult flies emerged from pupae and required several days of feeding before reaching sexual maturity. Quality control systems tested random samples from each batch to verify complete sterilization. Researchers mated sample males with fertile females and monitored egg development to confirm no viable offspring resulted. Any batch showing partial fertility was rejected entirely. The testing process required maintaining laboratory colonies of fertile flies for comparison studies. Quality control data tracked sterilization effectiveness across different seasons and production conditions. International agreements established protocols for cross-border cooperation in sterile fly programs. Countries had to coordinate release schedules to prevent gaps in coverage along national boundaries. Standardized procedures ensured consistent quality across different production facilities. Research sharing agreements allowed improvements developed in one location to benefit all participating programs. The realization that success demanded unprecedented coordination between scientists, governments, and military-style logistics operations marked the beginning of biological warfare on a continental scale. This wasn't pest control anymore. It was a sustained campaign requiring industrial production capacity and permanent commitment to maintaining operations indefinitely. The scale of infrastructure needed to wage this war would dwarf anything previously attempted in biological control, creating something unprecedented in biological control. The Copen facility in Guatemala stretches across 30 acres of industrial buildings that operate like a biological weapons factory disguised as a research center. Inside these climate-controlled structures, workers tend to millions of screwworm flies in various stages of development. The facility produces approximately 40 million sterile males weekly, requiring a production pipeline that never stops. Each building serves a specific function in the assembly line of death egg collection rooms, larval rearing chambers, pupation areas, adult fly colonies, and radiation sterilization facilities. Modern breeding operations bear no resemblance to the primitive meat-based systems used in early experiments. The original facilities fed screwworm larvae on chunks of rotting meat, creating horrific working conditions and inconsistent nutrition that produce weak flies. Today's operations use precisely formulated artificial diets that provide optimal nutrition for rapid development and maximum adult vigor. The synthetic diet contains exact ratios of proteins, carbohydrates, vitamins, and minerals that produce flies superior to wild populations in size and competitive ability. The production process begins with maintaining breeding colonies of adult flies in massive screened enclosures. These colonies require constant monitoring to ensure proper sex ratios, adequate nutrition, and optimal environmental conditions. Workers collect eggs daily from artificial substrates designed to simulate wound conditions. Each egg batch is tracked through the entire production cycle with barcode systems that monitor development timing, diet consumption, and emergence rates. Quality control begins at the egg stage with microscopic examination to identify any abnormalities that might affect adult fly performance. Larval rearing occurs in temperature-controlled rooms filled with thousands of containers holding developing flies. The containers are arranged in precise grids that allow automated feeding systems to deliver measured amounts of artificial diet at programmed intervals.